Hello everyone, today we will of course continue on this lecture of embryo development the first few weeks. Here we have the 4 blastomeres, 40 to 60 age, 8 blastomeres, and we have the 12, 16 blastomeres, formula 3 days. Before we have the unfertilized ovum, then fertilized Asian blastomeres 2 after half hour, zona pelicide 12 to 16 blastomeres, and blastocoil 60 hours. Following the sequence at the top left and cross and down to the bottom right. Fertilization occurs between 12 to 24 hours after ovulation. The release of an ovum from the ovary, fertilization occurs in the distal third of the oviduct. When an ovum is fertilized by a spermatozoon, the gametes, ovum, and spermatozoon each contain only 23 chromosomes. The haploid number when the egg is fertilized as shown, the chromosomes from each gamete meet in the nucleus of the zygote, producing 46 chromosomes. The diploid number 22 pairs of autosomal and 1 pair of sex chromosomes. The sex of the zygote depends on whether the sex chromosome of the fertilizing sperm is an X or a Y. The sex chromosome carried on the ovum is always an X. At fertilization, the zygote commences a series of mitotic cell divisions, cleavage divisions. These divisions will finally result in the formation of a new individual comprising millions of cells, all derived from one single cell. The zygote will give rise to all other cells in the body. Put another way, it has the potential to produce all cells of the body and is therefore totipotential. The original single cell zygote divides into 2, 4, 8, 16. Each cell is known as blastomere. The time after fertilization at which each stage is reached. The 2-cell stage attained about 30 hours post-fertilization. The 4-cell stage after 40 hours. 12 to 16 blastomere stage is about 3 days, and by the time 16, the balls, now called morula, has reached the uterus. If you have not noticed already, the 16-cell stage, morula, is the same size as the original ovum. It's a duplication of the reality. 16 blastomeres occupy no more space than did the original egg. As each cleavage division occurs, each new nucleus takes its shape of the original cytoplasm of the ovum. Only new nuclei and cell membrane are added at each division. Note in that the division ovum the dividing ovum surrounded by the zone pellicida, the dividing pole. It prevents the entry of the subsequent sperm once has penetrated the ovum and fertilized it. If the blastomeres, making up the 16 cell morula, were separated from each other at this stage, each one has the potential to form a separate embryo. Each blastomere is still totiopotential. These totiopotential cells are known more generally as embryonic stem cells and are the subject of intense ethical debate about their use in medical research. Further examination of the final steps of the journey of the fertilization of the ovum to the uterus shows that the morula continues to divide, but the cells now begin to segregate, those forming the inner cell masses in the center of the differentiated from those on the surface of the ball. The inner cell mass form the embryo, whereas the outer cells will form the trophoblast, from which the placenta will develop. The blastocoel is created as uterine fluid passes between the outer cells and the inner cell mass gravitates to one end of this cavity. The resulting structure is known as the blastocyte, which contains 100 to 150 cells. The zona pellicida has disappeared. Towards the end of the first week, at about 5.5 days after fertilization, the blastocytes becomes attached to uterine wall. As shown, attachment of the blastocytes is the first event in the process known as implantation, during which the placenta forms. The placenta is the organ where exchange of nutrients and waste products take place between the maternal and fetal circulation. Effects of theolytic secretion from the trophoblast. They erode the lining of the uterus, and cells from the outer cell mass invade the space created. The blastocyte thus becomes implanted The blastocytes, thus embedded in uterine wall. Now, sure. Yes. Okay, sorry about that. So we finish. As you can imagine, even in the apparently simple sequence of mitosis during the first week, there is a lot of scope for things to go wrong. These are described early embryonic abnormalities as accidents of fertilization are common, but their causes are poorly understood. It has been estimated that between 1 and 2 out of 3 zygotes develop an abnormal number of chromosomes or major mutations within the normal number of chromosomes as early cleavage division takes place. As early cleavage division takes place, most of these abnormal embryos spontaneously abort in the early stages of pregnancy, often without the mother being aware that an abortion has occurred. The loss of embryo appearing as a late period because so many abnormal embryos spontaneously abort. Because so many abnormal embryos spontaneously abort, only some 6 per 100,000 individual live births have detectable chromosome abnormalities. Probably the best known of these is the Down syndrome, in which are 3 instead of the usual pair of 21st chromosome, trisomy 33. Multiple pregnancies arise in 2 ways: dizygotic twins, or more, result from fertilization of separate eggs by separate sperms and are no more like than brothers and sisters. Identical or monozygote twins, triplets, or quadruplets are the result of the splitting of full zygotes into 2 or more individual blastomeres that each develops into a separate embryo. These embryos are the same sex and genetically identical. About 70% of twins are dizygotic, 7 per thousand births, and 30%, 3 per thousand are identical zygotes. Ectopic pregnancies produced when a blastocyte implants outside the normal area of the uterine mucosa. Sites where this may occur are the oviduct, ovary, cervix, and the uterus, and the abdominal or pelvic cavities. Such pregnancy almost invariably fail to go to term and may produce serious hemorrhage as the burrow into the surface of abnormal viscera, surrounds the blastocyte completely and large spaces surrounds spaces develop within is known as lacunae. By the 10th day, the lacunae have made contact with the maternal sinusoidal blood vessels in the uterine wall. The 10th day, the lacunae have made contact within maternal sinusoidal blood vessels in the uterine wall. The embryonic vacuoles and maternal sinusoidal vessels provide a large contact area between the two circulatory systems for exchange of nutrients and waste products, thus forming the basis of the placental circulation. Formation of the bilaminar embryo. While the placental circulation is being established, certain change takes place in and around the embryonic disc itself. If you compare figure, you can see how the inner cell mass, shown in A, differentiates into two layers, shown in B. As implantation proceeds, a layer of columnar cells, the epiblasts of future ectodermal feces, the cytotrophoblasts, and a layer of small flattened cells, the hypoblasts. The embryo briefly exits in this two-layered form as the bilaminar embryo. Examine the change around the embryonic disc itself. A loose agglomeration of cells of uncertain origin appears and spread along the inner surface of the cytotrophoblasts and surround the embryonic disc. These cells form the extra-embryonic mesoderm. At the same time as shown, the hypoblasts extend to form a cavity called yolk sac. The extra-embryonic mesoderm between the cytotrophoblast and the yolk sac form a loose tissue with large vacuoles that later unite to become the extra-embryonic coilicoma that plays a crucial role in the nutrition of the development embryo until a vascular system is established as the coileum forms a more solid area of extraembryonic mesoderm, the means which connects the embryo to the trophoblasts. This is connecting stalk, which is the first sign of umbilical cord. The third week, gastrulation. The third week produces significant change in the embryo. Two more germ layers, intra-embryonic mesoderm and endoderm, are added to the epiblasts in a series of processes known as gastrulation. Towards the end of third week, post-fertilization, the central nervous system begins to form during neurulation and fourth germ layer known as ectopic mesenchyme is produced. As it does so, all the basic building blocks are then placed different to specific cells that will aggregate into tissue and organs. Pregnancy testing. Fertilization usually occurs about 14 days before the next menstrual period is due. Considerable development has already occurred by this time. The embryo has reached the biliminar stage, and gesticulation is beginning. The usual signal that a female is pregnant is when the next period fails to occur. In many countries, pregnancy testing kits are available for self-diagnosis if pregnancy is suspected. These detect the presence of a hormone known as human chorionic gonadotropin in the urine. HCG is produced from the trophoblasts as implantation and placentation take place in the second week of pregnancy. The role of this hormone is signal to presence of the implanted embryo and to suppress the usual cyclical release of hormones that controls the maturation and release of ova at ovulation. In the second week of development, the major event is the implantation of the embryo into the uterine wall and establishment of placenta. Relatively little change occurs within the embryo, although some important changes occur externally as a prelude to establishment of the embryo in general. Placentation. As implantation takes place, the distinction between the trophoblast outer cell mass and the inner cell mass becomes more obvious. The trophoblast continues its erosion of the uterine wall so that by the 8th day post-fertilization, the blastocyte is firmly embedded in the uterine wall. The trophoblast differentiates into two parts as it works its way deeper to the uterine wall. Leading portion adjacent to the uterine tissue becomes syncytial trophoblast, in which the individual cells use their cell membrane to form a multinucleate syncytium. The cells adjacent to the embryo remain as distinct cells, forming cytotrophoblasts at the stage. The embryo proper is growing much more slowly than the tissues surrounding it. By the 9th day of development, the blastocyte is deeply embedded in the uterine wall due to the further enzymatic activity of the syncytiotrophoblasts, as shown in picture. Thank you for listening to this amazing lecture, and it is a part of let's read and enjoy this amazing book. Tomorrow we will also